Hello, my name is Brianna Blount. It is the 25th of January 2026, and the chiral molecule I chose is lysine. The IUPAC name for lysine is 2,6-diaminohexanoic acid. The chiral center in lysine is the alpha carbon, and that is attached to 4 functional groups. The 4 functional groups that are attached to the A carbon or the chiral center in lysine in priority order. The first group being the amino group, the NH2. The second functional group is the carboxyl group. The third functional group is there's a side chain as well, the CH2,4NH2 side. That extra amino group that lysine does have, it allows for it to form shift bases and isopeptide bonds as well. And the last group being hydrogen group. Lysine has 2 enantiomers, L-lysine and D-lysine. Again, the enantiomers are the non-superimposable mirror images, and yeah, the 2 for lysine are L-lysine and D-lysine. Lysine does not have any diastereomers. Diastereomers require 2 or more stereocenters, and lysine only has 1 chiral center, so that makes it impossible for lysine to have any diastereomers. And lysine has several constitutional isomers, being 2,5-diaminohexanoic acid and 3,6-diaminohexanoic acid. Due to its functional groups, lysine contains 2 amines and a carboxylic acid, and that contributes to the uses. Some of the lysine is useful for making acid-base reactions, zwitterion formation, pKa comparisons, and salt formation. And lysine is an essential amino acid, and we do not produce it, so it must come from our diet. Some foods that are high in lysine are cheese, soybeans, pumpkin seeds, eggs, and quinoa. Lysine is one of the 9 essential amino acids, and the 9 essential amino acids are amino acids that we do not produce naturally. So these are amino acids that we would need to intake through our diet. Growth and tissue repair, it's especially important in children. Collagen formation and calcium absorption. Another use for lysine is the cell culture media component, protein labeling and modification. Yeah, lysine essentially allows you to modify proteins. That extra amino group in the side chain for lysine, it acts as a common target for acetylation because it is a strong nucleophile. It also acts as a target for methylation. And these processes can regulate the expression of genes through epigenetics. Histone lysine modifications to regulate gene expression.